There it goes. Just wanted to make sure I could see the little ticker going before I get started. All right, of course, I've not finished all of today's lecture, so I don't know where I'm going to get to in the lecture, so I'll have to tweak the exam before we give it on Thursday to make it fit. But I do have a draft of the exam here, and rather than typing out a bunch of terms and giving them to you on Blackboard, I'm just going to record this as part of today's audio lecture and then post it on Blackboard so you can listen to it, and that saves me having to type anymore. I spent all day yesterday typing and retyping exam questions so that I could come up with this. Now, we're covered chapter 5, which is all about bacterial growth. Then we covered chapter 6, is kind of the molecular biology of bacteria. And we're trying to cover chapter 8, which is more regulation. We skipped chapter 7. See, I do that when there's a recording device around. I can't talk properly. We skipped chapter 7 because it's mainly talking about eukaryotic cells and archaeal cells. And quite frankly, you're going to get that elsewhere. So I skipped that chapter so we could cover a little bit more. The last thing that we covered from the genetics kind of fits really well into this molecular, and that was the PTS system. So you need to know the PTS system. Remember, the PTS system is the phosphoenol pyruvate phosphotransferase system, and it transfers phosphate groups down a chain of proteins, enzyme 1, histidine protein, which is HPR, and enzyme 2A, glucose, are the ones that I use. This is my simplified version of it. What happens is they're transferring those from pyruvate, which is a high energy molecule that you get out of glycolysis, and that provides the energy to lock glucose or preferred substrates into the cell. Once it's in the cell, it's locked into place, you've got glucose 6-phosphate. What happens is it can dephosphorylate that last enzyme faster if there's lots of it coming into the cell than the chain can rephosphorylate it. So it comes down to it, it's giving you a ratio of phosphorylated to dephosphorylated enzyme 2A glucose molecules. If you have a lot of the phosphorylated form, that means there's not much glucose coming into the cell, okay, because the phospho chain is caught up. And that's going to mean that I need to start looking for alternatives to glucose to use for food. So I'm going to do certain things with it. I'm going to turn on adenylate cyclase, which converts ATP into cyclic AMP, and that's going to allow me to regulate genetic systems. Remember, we've been talking in this last chapter about DNA binding proteins. You have the promoter, the operator sequence, and that sort of thing, but you also have some cap binding sites, is where the cyclic AMP will bind onto that, and it just makes it a lot easier to transcribe and make proteins from a specific gene. So even if you have the inducer coming into the cell, like say maltose is the non-preferred substrate in this case, if you have the non-preferred substrate coming into the cell, and you also have cyclic AMP, that means that the energy levels are lower, and it means that the inducer is present. So now we will turn on the stove to cook maltose. We will turn on all those genes necessary for that. That's catabolite repression. If you have a lot of glucose coming into the cell, it dephosphorylates the enzyme 2A, has a negative effect on adenylate cyclase, lowers the amount of cyclic AMP, and even if maltose is present in the environment, you don't turn the genes on at any high level because you don't have cyclic AMP to bind to that right behind that promoter region. Does that make sense? Okay. Then you also have inducer exclusion. That's a simple one. The dephosphorylated form of the enzyme 2A binds to the transport molecules of the non-preferred substrate and prevents them from entering the cell. Okay. And third is expulsion. You already have some maltose in the cell. Glucose becomes available. We want to spit the maltose out because glucose is kind of easier to digest. So we dephosphorylate the maltose using the HPR. Then it passes it to the enzyme 1, or enzyme 2A, and the enzyme 2A then passes it to glucose. So once it's dephosphorylated, that maltose molecule would flow out of the cell through its transporter. It's no longer locked inside the cell. All right. So that's the PTS system in a nutshell. A lot of what we did in one of the chapters was talk about, in all of these, we talked about making polymers. We made polymers of DNA, we made polymers of RNA, and we made polymers of proteins or amino acids to make proteins. So you need to know a little bit about DNA replication, transcription, translation, and those three major things will cover a large chunk of the test. Okay. Chapter 5 is pretty easy. It's talking about growth curves, that sort of information. Let's see. You're going to talk about the growth cycle, lag, log, stationary, and death, knowing that you're doing that on like logarithmic paper in order to see that. Know what the lag phase is. The lag phase is where you turn on different genes. It's an adjustment period. When you move a bacterium from one growth media into another, you're going to have a lag period. Even if you're moving it from the same kind of growth medium into fresh growth medium of the same type at the same temperature, you're going to have a lag period. It's just because waste materials build up in the media, and it's going to be a different chemical composition in an old culture than it would be putting it into new. So you still get that kind of lag phase. If you're working in industrial microbiology, you want to reduce that lag phase as much as possible to keep the bacteria producing whatever chemical you want. So continuous culture becomes something that you can do. Now, there are different ways of measuring the number of bacteria. You can do plate counts, pour plates, spread plates. Those are not really accurate, super accurate measurements. They just give us a kind of a rough estimate. That's one of the problems with doing micro is sometimes the bacteria stick together, and that's one colony forming unit. So we're counting colony forming units, which just gives us a good estimate of the bacteria that are there. Now we can use turbidity measurements where we're using a spectrophotometer to measure the growth rate of bacteria. And when we use the spec, that's fairly accurate on growth rates. You can get pretty accurate numbers because the more bodies floating in the liquid, the more turbid the liquid is. So you can measure that using light waves. We talked a little bit about biofilms in here, what planktonic, what sessile bacterial cells look like. We talked about some of the different growth conditions of bacteria. You should know the terms that go along with them, like a psychrophile is one that grows in cold. Kind of know the range of temperature in which they grow. Know what some of the extremophilic ranges are. So when basically read chapter 5. Some bacteria can even survive in boiling water. Most of those are archaeal groups, but they can survive and grow in boiling water. It's pretty amazing, isn't it? Still blows my mind. A lot of those are in Yellowstone National Park, and they have so many solutes in the water. There's so much other stuff in the water. Calciums, magnesiums, all sorts of ions are in there that it allows the water to get above the boiling temperature before it kind of bubbles completely out. Now let's see. Know what prokaryotic genomes look like. They're double-stranded circular DNA, but you can have plasmids serve as extra chromosomal DNA segments. So you can have a bacterial cell. They're basically haploid, meaning they have one copy of each gene, but they can have some diploid characteristics if you put plasmids in the cytoplasm that have the same gene on them that you would find on the chromosome. DNA replication is kind of a bidirectional process. Remember, you've got to open up a large replication fork. You've got to so you're going to have your helicases open up that supercoiled DNA and widen it out, and then you have single-stranded binding proteins lay down. And here's what's important. DNA polymerase can't bind to a single strand. So what do you need? You need to lay down a primer. So on the leading strand, you're going to lay down a single primer. And what is the primer made of? RNA. RNA. So it's an RNA polymerase. Sometimes you'll see it called a primase that lays down the primer on that leading strand. And you know, they may be 15 nucleotides long. What direction do you read the template DNA? 3 to 5. You read it 3 to 5, and you make it 5 to 3. Don't forget that, and try to keep up with where the strands are located. Now, once the primer's laid down, a DNA polymerase can bind and start chugging along. As long as it's chasing the directionality of the opening of the replication fork, it doesn't need to lay down more segments. But if you're looking at the reverse strand, then you know you've got the reverse strand is opening up behind it. So you have to lay down multiple primer sets in order to be able to replicate. So you get the Okazaki fragments that are formed. Okay. And then all of that will have to re-anneal and reform. I think you've heard that until you're sick of hearing me talk about it. Ribosomal RNA has a function. Oh, you ready for me?
If you're ready for
Come on in. Come on in. Do you mind me recording?
I don't know what you're talking about there, but ribosomal, did you say RNA or DNA?
Ribosomal, well, it's ribosomal RNA forms the ribosomes. And the ribosomes actually are where you make the protein, and it's the only one that has a protein component. Remember that? What's the protein inside the ribosome? Come on, don't embarrass me.
Got me.
What's it do? What's the ribosome do? It makes the protein. So the protein that's in the ribosome
Makes the phosphodiester bond.
there. Thank you.
Whoa. All right.
Or
I'm
actually, it's
impressed.
a peptide bond.
Obviously, I need to go back to school. Thank you, Dr. Blair. Very quickly, I know class time is very valuable, and I'm just going to take a few minutes. But let me introduce myself. I'm Charles Ousley with the School of Pharmacy at Auburn, and I'm here really today to share with you about 3 things that maybe you want to drop by and spend some time. I'm going to be up in 216 in the conference room for the rest of the morning and into the afternoon if need be. This is Jacksonville State Day. So with that being said, what 3 categories am I looking for? Number 1, if you're honest with yourself, there are some of you in here that do not have the foggiest idea what you're going to do with the rest of your life. You know, you're thinking about pre-health, you're thinking about other careers. You know, if you've never stopped and thought about pharmacy and need some information, feel free to come by and just talk to me. I am a pharmacist. I went to that other school in the state, Samford University. It is a great school of pharmacy. I went to University of Tennessee to get my doctorate degree. But then most of my career has been in hospital pharmacy, independent pharmacy, nursing home pharmacy, so I can talk to you about the career. I can talk to you about what to expect. And I've got some information I can share with you about the career. It's no longer just retail and community. There's so many different things you can do with your pharmacy degree. So think about it from that perspective if you have a moment today. And then secondly, some of you are pre-med, pre-dent, pre-fill in the blank, and that's okay. We need those professions as well. You know, not all of you are going to be admitted to those schools. Not all of you that might like to get into pharmacy school will be admitted. So think about having an alternate plan and take a look at pharmacy from that perspective. If I didn't get to be a blank, you know, would pharmacy still satisfy my needs? And then thirdly, those of you that know you want to go to pharmacy school, that are looking at applying within the next several years, certainly from my perspective, these are the people that pay my check. You know, that's why I'm here today. I can talk to you about being admitted to our school. You know, with that in mind, guys, there's about 4 criteria that it doesn't matter whether you're a doctor, whether you're a dentist, pharmacist, we're all looking at. What's number 1? What do you think the first thing we look at is? Yeah, that's right. If you don't have the brain power to be successful, you know, then it's not going to happen. So we're going to look at that GPA, especially in courses like this. especially from schools like this. Do you have your degree? So academics comes into play. You may say, gee, I don't know why I'm taking this course. I'll never use it. Well, guess what? Somebody in a professional school is going to look at it from that perspective. You know, number two, you're going to have to take some kind of standardized test. You know, it could be MCAT, it could be PCAT, it could be OAT, DAT. We're going to look at your biology and chemistry scores more heavily than anything else. So you're going to have to take a standardized test. Number three, you better be doing now, and that's your application. You know, are you working? Are you job shadowing? Are you gaining experience in your respective profession or school to which you would like to go? You know, what organizational responsibilities? Do you have to work full time? You know, there's nothing wrong with working. I had to work, not a problem at all. And that's going to impact all those other criteria. Where do you do volunteer work? We know you got free time. And y'all are going to say, we don't have free time in college. Trust me, you'll never ever have as much. But you got to experience that for yourself, and I understand that because I did the same thing. What is Dr. Blair going to say about you when you ask him for a recommendation letter? Is he going to highly recommend you? You know, you want that to be a favorable recommendation. So those three things I tell students, you know, guys, we're going to make a decision on your future with our school, and all we have seen is paper, paper, and paper. Okay? The fourth thing is the interview. So take advantage of Career Services or Career Center here at JSU. Nearly every Career Center will offer a mock interview for a professional degree. You know, if you're serious about applying to a specific school, see if they will give you a mock interview. Polish your interviewing skills. Critical nowadays. So those four things, basically to some degree or the other, some schools will weigh one of those attributes more heavily, and that's your responsibility to find out. But having said that, those are the four big things that get you into a professional school. So as I said again, I'm going to be upstairs, and I've taken, you know, this day for JSU, you know, this morning, this afternoon, whatever time I need. If you have a moment and want to drop by, I've got information about pharmacy as a career and certainly information about what you need to do. So with that being said, I'm going to turn it back over, but who's your favorite professor? Who said that? Brian Stroud. There you go. Have a good one.
Now, how many of you are pharmacy again? Hands up. Look, there's quite a few pre-pharmacy people, and I'm giving them permission if they want to.
All right, the price is right. We're not going to come on down. We're going to come on up, okay?
All right. He's got a lot of good information on what it takes to get into their program and the application process and that sort of thing. So I'm glad that he's able to shorten that into that quick process so that we can get back to covering for the test. But if you want to go up and talk to him, you're more than welcome to. I am recording everything. Make sure it's still, yeah, it's recording. And I will post it on my YouTube link so you can get to this. I'll have it posted today. Let's see. DNA replication. What is in DNA replication? Well, we kind of already covered it. You're making copies of the whole thing. And that's a big process. It's a lot bigger than, say, transcription. Transcription, you only need to open up a small transcriptional bubble, and you're going to be reading one strand, which is called the sense strand, and it's reading 3 to 5, and then it makes it 5 to 3. So as the 5' end come out, the one that's different is when you have ribosomes binding onto that messenger RNA. Now, what 3 kinds of RNA did we make in the cell? Messenger, transfer, and ribosomal. Those are the 3 types. You need to know what they are and what they do. Let's see. We'll have been talking about the lactose operon, and I will definitely finish that today. We talked about catabolite repression, growth, what environmental factors can affect growth. You've got oxygen levels, so you've got strict aerobes, strict anaerobes. You've got facultative anaerobes, which can grow either in the presence or in the absence of oxygen. So how do oxygen levels affect the growth of bacteria? Yeah, I've already mentioned you need to know all of those terms that are associated with them. An alkalophile would grow at what pH range? What are more basic, but you might want to look and see what the ranges are. Organisms live in environments. Okay, you've got halophiles, you've got osmophiles, you've got xerophiles, aerotolerant bacteria, which are really anaerobes, but they can handle a little bit of oxygen for a short period of time. There's all sorts of terms that are associated with that chapter 5 that we covered. Let's see. All right. Peptidoglycan biosynthesis is fairly complex. I didn't go into a lot of detail on it, but there are some drugs that affect cross-linkages and the way that peptidoglycan is made. And bactoprenol is one of the ones that you might want to pick up on. We've covered a lot in these chapters, and like there's the CRISPR system that you need to read about. I talked about that last time. Trying to think. No, don't forget that a cistron is actually a gene, so a polycistronic messenger RNA would be a messenger RNA strand that codes for multiple genes, which means you're going to get multiple proteins from a polycistronic messenger RNA strand. That's just, I don't want you to get confused. I could say many genes, but they call it polycistronic. I don't know why. Be able to draw a DNA molecule. Be able to understand where the 3' and the 5' end are located on a long chain of DNA.
So we have to draw the carbon skeleton and all that?
Yeah, I mean, just roughly, if you can draw a ribose sugar, a base, you don't have to draw the intimate details of the base, but you can put A or T or C or G there. Know that there are 2 hydrogen bonds between the A and the T, and there's 3 hydrogen bonds between the G and the C, and they're located in strands that are running anti-parallel to one another. I want you to be able to recognize or draw that double helix. If you were to cut a strand out, this is what this chain looks like, this is what this chain looks like, and put them together. It's kind of like a puzzle. It's fun. Let's see. Know the ribosome. So when you're talking translation, know how the ribosome works. Where the transfer RNA comes in, it dumps into place. What's the first amino acid in prokaryotic cells? I'm sorry? Well, that's the first codon. What's the first amino acid that it codes for? It's that formulated methionine. Okay. Yeah, so you've got your first codon that goes in the P site is always that formulated methionine. Then the 50S subunit kind of binds, and then the next transfer RNA will come in and put the amino acids together. Know or be able to recognize a peptide bond. I may just hand draw it. It doesn't have to be pretty, just so you can recognize what's there. And I may draw 2 or 3 chemical reactions and have you pick out the right one. Or better yet, just know how to draw a peptide bond and how it forms. Boom, then you got it. Okay, it applies to all of the amino acids across the way, and it's just a dehydration reaction that causes that peptide bond to form. How does the ribosome fall apart at the end of transcription? We talked about some ways that bacteria can pick up genes from other bacteria. I don't know if you remember that. It was way back at the beginning of chapter 5. We talked about conjugation, transformation, and transduction. And I went completely blank on the Griffith experiment. I couldn't remember it was called the Griffith experiment that day, but that's what it's called. That's the one where they took the smooth and rough colonies of bacteria, the smooth would cause the mice to die. If you killed the smooth with heat, the mouse lived. But if you mixed a rough and a smooth together, then you would result in transformation of the rough into the smooth form. So it would then kill the mouse. So the Griffiths experiment is something you'll want to look up. It is kind of cool in here. I look over there and she's got that thing wrapped around her head. I hope she doesn't, just all you see is a nose sticking out. We talked about regulatory proteins and DNA replication. Remember that a lot of the regulatory proteins are going to be binding into that large groove on the double helix that's there. So as they bind into place, a lot of them will bind in and then form some sort of bond. It doesn't necessarily have to be a covalent bond. It could just be van der Waals forces or whatever. And that will prevent that segment of DNA from being able to unwind properly. So as long as those regulatory proteins are in place, they stop the transcription and translation. What is enzyme induction? Enzyme induction, anybody? From freshman biology? Maybe you should look that one up. How do you turn on enzymes? Enzymes can be regulated in multiple different ways. So how do you induce them? I'm sorry? That's one way. Yeah, that's a catabolite repression. Can actually be used as an inducer for the production of certain enzymes. Some enzymes are produced, but they're not activated until they reach certain pH. Remember, we talked about peptidoglycan, how the enzymes have to move through and then chop the old peptidoglycan. You don't want them working near the surface. So if it's more acid, there's more hydrogen ions near the surface of the cell. They've got to migrate through all of that peptidoglycan before they become activated and cause the clump of peptidoglycan to separate. What's a transposon?
Genes that move.
Genes that move. What's an IS element? Insertion sequence. It's usually an inverted repeat that sticks into the genome somewhere. But you do have genes that move around on the chromosome, and they can move from the chromosome to those cryptic plasmids. Anytime you see the word cryptic in microbiology, all cryptic means is I don't know what it does. So it's just a much fancier way of saying, I don't know.
It's a mystery.
But it sounds much more impressive if I say cryptic than it does if I say I don't know. It's like if you're looking under a microscope, there's the word artifact that's used frequently. And artifact it's some crap I can't identify. Literally. So it just sounds better if you're looking at, there's a lot of artifact on this slide. That's like saying there's a lot of crap on here I can't recognize. It doesn't have any particular shape. Microbiologists do like to do that, come up with terms that sound better than saying I don't know. If you ever do fecal smears, you'll know what I'm talking about. With a fecal smear, you kind of have to take your eyes out of focus on the lower powers, and you're looking for something that is too organized to be crap. You're looking for something that is perfectly round or perfectly oval. It's kind of got symmetry to it, and that's what you focus in on. So you kind of have to take your eyes out of focus, and all that stuff that's just garbly-gook in the background you ignore, and you look for the things that are kind of symmetrical. You do fecal smears if you're looking for parasites, and we'll get to a parasite chapter later on. They're kind of cool. Okay. Ooh. I've got essay. Answer all three of these, but I only have two written so far. So I've got to come up with a couple of them. One of them goes back to the genetics. One of them was a question posted in this review. I don't know what my third one should be. If you've got any suggestions, you can let me know. Maybe you should.
It should just be two.
Y'all are good. I like that. You make me laugh in the morning.
If you make it out of here.
If you make it out of here.
Yeah, it's so cold.
It's all right.
My door was shut this morning. Yeah.
Do what now?
Our door was shut this morning.
On your car? Ooh. Where'd you come from?
I didn't care. I said, I'm coming to class.
See, I'm so impressed about that. You need to remind me when you're asking for a letter of recommendation. I came on the pseudo snow day that we had that, it's okay. One of the reasons I'm recording this is because so many people did have kids and their schools were closed or delayed opening and they couldn't come. It's funny, I've got a lot more people showing up today than I did last Thursday. I mean, I see some faces in here and I'm like, have I seen that person before? Hmm?
The day before test day. They
It's,
listened to the review.
oh, okay. Well, that's great. I won't do this audio recording of the test review before every exam, so show up every time the day before the test day. All right, it's time for us to get started on the lectures. Okay.
And then the stuff we're covering today is on the test, right?
Do what now?
The stuff we're covering today.
The stuff we get through today, I will add to the test. I'm still not through with the test. That was the draft that I finished yesterday at 4:30.
If you want, you just send me rough draft of your test, and I'll look it over for you.
Yeah, I could post some rough drafts, but they would probably scare you to death. Way back in the day, I would give a total discussion test for this. And yeah, that's back when I wasn't married and I had no life, so I graded all that crap. Now I'm learning. All right, let's see. I don't know why that's cut off at the top. Oh, it works better if you turn it on. But that cut off, see it says positive control up there?
This is a sign.
It's a sign?
Yeah.
Okay. It's a sign that we need to look at the TV.
No, we need to get out of here. It's so cold. God.
Well, shivering will help warm you up a little bit. Those people that are from the north can really laugh at us down here.
No, I'm from the north, and I still don't like the cold. Hey, you adjust after
You adjust?
Some babies.
Oh, okay.
You should go into lab and tell a Bunsen burner.
A lot of us in the South don't know how to dress. Like I noticed you got on a t-shirt underneath that jacket, and you dress in layers, and it keeps you warm. Of course, I've got so much insulation, the cold doesn't bother me anymore.
Well, I didn't expect them not to have the heat on.
Yeah, it's like
cold
you dress in
front.
layers and you go inside, then you have to go like, oh, it's hot.
You never know how to dress for this place. You really don't. The next time we come in, it'll be 120 degrees or something. All right, so we're going to talk about positive control of transcription. There are a lot of regulator proteins that activate binding of RNA polymerase to DNA. So, positive control of transcription. That means that something binds to that railroad track of DNA or RNA, whichever is the case that you're talking about. It binds the RNA polymerase to the DNA. So the DNA is laid out. We're wanting to transcribe it into messenger RNA. If it's highly regulated, you will need to bind multiple things to that. So you can have proteins that bind to it. And to me, I see it as the airplane comes in for the landing. It can't land if that roadway is buckled. Well, if you add a protein to it, it straightens that out and it allows it to come in and land and go. So there's a lot of these positive control mechanisms. Maltose has an operon, and it's in the E. coli's the best studied. E. coli's been completely sequenced for quite a few years now. It was the first bacterium to be completely sequenced. And the maltose operon has activator proteins that cannot bind to DNA unless it first binds to maltose. So you've got to bind the DNA but you don't want to bind the DNA unless maltose is also present. So if maltose is present in the environment, it gets transported into the cell because you have some low-level constitutive production of those transporters. You bring maltose into the cell, it binds to the activator. And once that happens, then the activator can bind to the DNA and cause that roadway to straighten out. So now we can turn on the genes for the maltose utilization.
So the activator for that gene will only activate when maltose connects to the activator because it won't before.
Right.
Does that protein, will it bind to other sites if other stuff is
No.
available in the cell? So that protein is strictly for,
Strictly for the maltose alcohol. Yeah, and there's low levels of these proteins floating around in the cell for all the regulated genes. So when you run, if you were to lyse the bacteria and run them out on a gel, it's going to be just basically a smear of lane. And some of those bands will have 2 or 3 proteins that are together just because they happen to be the same size. So just running out a single gel is not going to even be close to separating all the different proteins. Some of them that are in low levels, like the really rare genes that are turned on, may only have 2 or 3 copies of the protein floating in the cytoplasm. So they're really hard to detect. Have you heard of 2-dimensional gel electrophoresis? 2D gels? 2D gels are really cool. I'm trying to think of a 3rd dimension for a gel, but I haven't come up with anything yet. So a 2-dimensional gel is pretty simple. You run it in one direction. So you take a tube, you load your protein, and you run it out based on the isoelectric focusing point, which is, it runs to a specific pH and it stops. So you get bands that are separated based on their isoelectric focusing point. Some of these bands will have multiple proteins. Then you take an SDS page gel and you run it out in the opposite direction. So what you wind up with are really separated proteins. Some of them will be close to each other. And a few of them will have multiple proteins in a spot. But you get much better separation. Now, if I want to do a process called proteomics, I'm working backwards. I want to find the protein that's useful. So what I do is I run a 2-dimensional gel on glucose. Then I run another 2-dimensional gel on cellobiose.
what do you mean you run the gel on glucose?
So let's say I grow up Eubacterium cellulose on glucose. I lyse the bacteria and I run the protein on the gel.
So you just feed it a bunch of glucose?
Yeah, you grow it on glucose. That means all the genes necessary for glucose utilization will be turned on, and the other genes will be repressed. Now, I go back and I grow the same bacterium on cellobiose, lyse the proteins and run it out. So now, those X's represent proteins that show up in the second gel that didn't show up in the first gel. Those are proteins that are turned on by cellobiose that are repressed by glucose.
Will the circle show up when you do this celluliose one, or do you just compare the two?
If they're constitutively produced.
Oh, okay, so that's how you find out if they're produced at the same time.
Right. So they'll show up. I mean, you'll have some that show up in anything you grow it in or any condition you grow it in, just because they're constitutively produced. There'll be some spots that disappear from glucose utilization to the cellobiose degradation. Usually not many, but a few. So now, I can identify that these X's represent some protein that is unique to cellobiose utilization. So I have a function for the protein. If I go in and I pull genes out at random from that genome, all it's going to tell me is A's and T's and C's and G's. I don't know what the heck it does. But by working this way, I can take that, get an sequence. An internal sequence on the protein, and I'll sequence 10 or 15 amino acids. I get that 10 or 15 amino acids, and I can create a primer, and I can go in and amplify the gene for that protein based on the internal sequence. So now I can study that gene separately. Okay, I can study just that one thing. And if you do that for all of those new ones, then you can actually get a pretty good profile of what's needed for cell bios utilization. They've got this great, one second, I'll let you ask your question. They've got this great computerized robot that you load your sample in there, and it will run 2 gels. It will pick the spots that are different based on which one you put in first. And then it will give you the internal sequence for all the proteins. And it's just this robotic arm that goes in and it picks the spots for you and everything. It's really kind of cool. Yes, ma'am.
What does it say about this? Like when you go this way first and then you go down, when you go down the second one, what does it say about the size of the proteins?
That's a good question, and I should have said something about that. That is based on size. This is a regular SDS page, so it's separating the size of the proteins. The larger proteins are not going to be able to travel as quickly, so they'll be near the top. The smaller proteins will be down near the bottom of the gel.
So the smallest proteins are all the way over and then all the way down, or
Well, this has nothing to do with size up here. It has to do with isoelectric focusing point. So the smallest ones will be across the bottom.
and this is not like the jelly matrix you described last week, right?
What
For the how we found out the 50S, what size is the 50S?
No. This is a polyacrylamide gel electrophoresis. If you were to take and you pour a gel, basically you mix some chemicals together that will polymerize into a jelly, and you pour it on a plate, what you get is a thin layer. Just imagine you've got a thin plate and you pour Jell-O in there and stick it in the refrigerator. So if I'm looking at it from the end, you get it. I can't even do it today, but what's going to be happening is there's little small pores, and the pores run throughout this thing. So a molecule hits one of the caves, and it kind of weaves in and out of all those pores as it moves through the gel. So the electrical current will carry it through there. The larger molecules have a harder time squeezing through, so it takes them very short distances. The smaller ones can squeeze through a lot more of the small holes, and they travel further in the same amount of time.
And what is IEF again?
Isoelectric focusing. Isoelectric focusing. I didn't even mean to get into 2D gels, but this is kind of a cool concept to throw out at you. It brings in that proteomics. They have a way of doing something called subtractive hybridization, where you take a cDNA library from the same organism grown in 2 different conditions. So you're grown in glucose, you're grown in cellobiose. I make a cDNA library. What's a cDNA library? Notice I made a note there. Might be a hint. I'm pretty sure I covered this. Okay. A cDNA library is where we make DNA from all the messenger RNA in the cell.
Is that the recipe? When we talk about the recipe and then those photocopies and then inoculate in the library? You don't remember that, do you?
I remember the analogy, but I'm not sure if I applied it to a cDNA library. You got a recipe book
Yeah.
in the kitchen.
Oh,
This
yeah.
is it, yeah.
Okay, so if you've got a recipe book in the kitchen, that's the DNA. But the kitchen is only cooking about 20 of these recipes. The recipes are messenger RNA floating around in the cell. So now if I go in and I convert all those recipes back into DNA, that's a cDNA library. So I isolate the messenger RNA. I sequence the messenger RNA, and that represent what genes are turned on, not what genes are present in the genome. So I've ruled out all the genes that were not turned on at the point in time that I made my cDNA library. So now, you have proteins and you have proteins, and a lot of the proteins are similar, but we've made a cDNA library of all those proteins. So now, subtractive hybridization means that I take and add something heavy to the DNA in this cDNA library. I label it with something. Okay. I can't remember what it is. It's gold or something. And so your double-stranded DNA fragments that are floating in the cDNA library are heavier than hers. So I mix those two together, I heat it up, and I allow hybridization to occur, which means like binds to like. So if you have the same gene she has, some of those heavy DNA molecules will bind to some of her light ones, and then you spin it down and separate out the ones that are too heavy. And I've now subtracted all the common DNA fragments, and all I have left are those rare, unusual DNA fragments. That's subtractive hybridization.
We figured that out. Like, how do you even come up with that?
You know, it's kind of fun to sit around and try to think of new ways of doing stuff. Anytime I read an article, I think, how can I do this? How can I use this? You guys need to be reading Science Daily News, Mortality and Morbidity Weekly. If you're going into pharmacy, there's got to be some pharmacy weekly thing that comes out. Sign up for the free newsletter that comes out daily or weekly or whatever it is, and you can just read the headlines. You'd be amazed how many times you read a headline and then you go, "Whew, let me read the abstract." Well, then later that day, somebody will ask you a question about something you just read that morning, and you sound a hell of a lot smarter than you really are. But it lets you know what's happening in the world. If you're going into nursing or medicine, you need to know what diseases are coming your way. You really need to know, oh, there's a new strain of tuberculosis that's out, and it's eating through the plastic and choking you to death. You know, it's a bad one. They don't really do that, you know. They're a little slower to get in there, but you get the point. You know what's happening in the world, your world, and so sign up for some of those newsletters, especially if you're determined to go into pharmacy. Ask Dr. Woolsey if there's a weekly or a daily newsletter that comes out. Read the titles. You'd be amazed how much you learn from just trying to. You don't even have to read the whole article. Sometimes just reading the title and maybe trying to figure out the abstract if it's something that jumps out at you. You know, different things like snake venom usually catches people's attention. So you read about snake venom. They've isolated, with a cDNA library, they've isolated a painkiller from snake venom that also binds to the same receptors that heroin binds to in a different way. So they're looking at this particular painkiller as a way of treating heroin addicts to cause them not to have, you know, the.
They could have given it a really cool name, like you know, based on snake venom, you know.
I know, but see, I don't like the idea of injecting myself with anything that came from venom or toxin, like Botox. I'm just going to be wrinkled because you're not injecting me with anything that's got the word toxin associated with it. Yes, sir.
Then if they use that snake painter, when they just package it TBD,
No, they could back off on it. The thing is, it wouldn't trigger the same high that heroin does. So that's the beauty of it, is it's binding to the same receptor and it blocks heroin from being able to bind to it. So if they started off at some level, then they can gradually lower the dosage, and it would be a way of treating that addiction.
Does it cut off on the pain of the withdrawal?
It replaces it almost.
It replaces it, but I don't think it triggers it to the same degree. You don't get the addiction. I read a title and a short abstract, so don't quote me on any of this. That would be Dr. Murdoch's area of expertise.
But it's pretty much like methadone, except not addictive.
Yeah. correction fern that allows it to withstand dehydration. Now they're trying to work on taking that same chemical and putting it into corn. So now you can literally plant corn in the desert, and when the rain comes back, boom, it continues where it left off. Wouldn't that be cool? You never know.
Ingenious, like, it seems ingenious when you hear things like that, but then for you to just sit down and think, like, you're like, why didn't I think of that?
You can. You can. Somebody may have beaten you to it, but just, I think about, do you know what northern, western, and southern blots are? These techniques are the things that are going to really save you. The first blot to come up was a southern blot. A guy named Southern came up with a way. Let's see.
Was that his name?
That was his name.
Oh,
That's
it's not
first and
about
last
north or
name.
south, it's about his name?
Well, what happened was DNA, RNA, and protein. So Southern came up with the DNA. A northern blot was a joke because the first guy was named Southern. They named their blot a northern blot, and it was RNA. And then a western was just continuation of the joke. It's using protein.
Is that a true story?
This is a true story. And that's how I remember it is snow and then the central dogma of biology. So what you do is you run a gel, you got your DNA bands in there. You take that band and you blot it on a piece of paper and you get exactly the same pattern again. So now you transfer that DNA to a nitrocellulose pad. And then you stain it with an antibody or some stain. So I know I'm looking for this gene. I can stain that gel, and the only thing to show up will be that one, maybe a faint one over here where you get some cross-reactivity. So I use an antibody or some stain that's specific for a gene that I'm looking for, and I can find that gene. Works for genetic engineering, works for testing. She's got severe diarrhea. I want to see if that, yeah, I'm sorry I gave you diarrhea this morning. Yeah, it's the stinky, it's nasty. So if we know a gene for that particular bacterium that we're looking for, C. diff, then we could run this, blot it, and we got a positive ID. That means from that total mass of diarrhea that we processed in the lab, you've got the gene in there, which means you've got C. diff. So we can tell whether or not the gene's present.
So it wasn't the tacos, it was the Geninos.
Well, it may have been the antibiotics you had last week.
So how do you treat people? Like, what do they do?
There's not a whole lot they can do.
Like, does it run its course and leave, or?
Well, it's not gone, it's still present. Anytime I see the digestive system as a stream environment. If you have a flood that washes everything out, then that can leave some bad stuff behind. So if you treat with antibiotics, you wash out the good things, the bad ones are left behind, and it can be a problem. Sometimes eating yogurt, taking antibacterials will overwhelm the C. diff. So a lot of hospitals now will give you small containers of yogurt after any antibiotic treatment, and that's reduced the number of cases of C. diff dramatically. Something as simple as that. Drinking buttermilk would also work. No? No?
It's an acquired taste.
I had somebody on Facebook this weekend posted a picture. It was a nurse, and she said to reduce your chances of getting influenza in the hospital, you need to swab triple antibiotic cream in your nasal passages. Remember, I had that happen on the airplane years ago. First, if you get your health information from a social media site, you should be beat with a wet noodle, okay? Second, antibiotics do not affect viruses. Antibiotics have no effect on influenza, so no. Third, there's a reason that the triple antibiotic cream says for topical use only. keratinized dead tissue is not killed by the antibiotic, so you're really just preventing infection at the site. You put it inside the nasal passages, there's no keratin, it goes in, it binds to the 70S ribosomes in the mitochondria and kills the cells in your nasal passages, making you more prone to catching a disease, not less prone.
I had no idea. Those are topical.
Topical surface.
I mean, I know what that meant, but why?
Some antibiotics can be taken internally without causing problems. If you wanted to kill yourself in a horrible way, you could eat a bunch of that triple antibiotic cream, and it would destroy the stem cells in your bone marrow and cause you to get aplastic anemia. Yeah, that would be. Yeah. I like to sit around and think of ways. I write novels with my girls, and we come up with some horrible ways to kill people. Because we do murder mysteries and that sort of thing. One of my biggies is somebody knocks you out, puts you inside the autoclave, and you wake up and you hear the buttons beeping. That scares the stew out of me. So, if you ever sat down to do a murder mystery, I know if somebody looked at my search history on the home computer, because we're always looking for different poisons. How would you make that poison from scratch, And there's digitalis, so you go buy a foxglove at the local garden shop, and then you dry the leaves and powder them up, and you can cause somebody's heart to stop. All right. Yeah, let's move on. All right, activator proteins bind to the specific sequence, and these are called activator binding sites, not operator regions.
Is that on the protein or on
The DNA. DNA. All right, so you can see it up here. Here's the DNA. There's the promoter region. There's E, F, and G, which are just proteins. So these are proteins that are necessary. The maltose activator protein is going to bind somewhere here, and this is in the activator binding site.
Is that whole thing, is that the maltose operon then, or is it concentrated?
It's starting it. So see, what you have is an inducer. So that means maltose is present. It binds to this maltose activator protein, which activates the activator and allows it to bind to this activating binding region. Excuse me. If no maltose is present, it's not going to bind. So once it binds, then the RNA polymerase can come in and land. The promoter would be right in there. And then it will start going in this way, and you'll get transcription of that particular operon. Promoters are positively controlled operons, and they only weakly bind to RNA polymerase. Activator proteins help RNA polymerase recognize the promoter. So activator helps the RNA polymerase recognize the promoter, so now we can make the messenger RNA. May cause changes in the structure of the DNA, may may interact directly with the RNA polymerase. So some RNA polymerases have this little groove. It's a site where these promoters will bind to it. So if you look at all the promoters for all the different operons inside a particular cell, there may be a sequence of about 20 nucleotides or 20 amino acid fitness protein here that are going to fit into that groove, and that could be a common feature to all of the regulatory proteins, simply because structurally it needs to bump up and bind to that RNA polymerase, and it bumps up in that same location for a lot of these regulators. So one thing that computer systems are doing now is they can search the amino acid sequence, fold the three-dimensional shape of the amino acid sequence that they've gotten from these proteins, and then categorize the proteins into groups based on common sequence similarities. And in this case, that one region, if that region is always present on those activators, then they would dump them into a box and say, all these puzzle pieces look like the edge. So now we can pull them out and study them separately. we used to have to do this by hand. I would get gene sequences, and it was kind of easy. You know, I know people that got PhDs by studying one gene. Today, you wouldn't get a PhD for less than a genome. I mean, it's changed that much. First human genome, $1.6 million to sequence. I can sequence a human genome now for about $1,500 to $2,000, depending on the group that I send it to. It's pretty amazing at how quickly we're advancing through these things. Is time up? Hey, no, we got plenty of time. Activator binding site may be close to the promoter or several hundred base pairs away, just depending on what's on this side of that binding site. So this is going to be binding on the RNA, but you can have the activator binding site could be way down the way, and this could be a long protein or it could be a short protein, but that would be the sequence that you would look for to categorize them. All right, so what we have here is we're forming a loop. So we've got the activator binding site up at, well, you can't even see it. There's an activator protein that's up near the top on the yellow over here. And it's got the RNA polymerase. And then we've got another activator protein down here. And you form this stem loop structure between the two. So then you can get transcription proceeding. If it's linear, if that's not bound there, you don't get the right three-dimensional conformation. So the DNA is not going to be laying right and the plane can't land. Genes for the maltose are spread out over the chromosome in several different operons, and each operon has an activator binding site. Multiple operons are controlled by the same regulatory protein, and that's called a regulon. Regulon. That's. I want to study global regulation in cellulose degradation. I want to have time to sit down and figure that puzzle out. If you could do that, you could be a rich person, because if you could turn on the genes for cellulose degradation constitutively, you could become rich. Industrial microbiology is all about regulation of the things that they have in their systems. There's a algae that produces an oil droplet. It does it in response to nitrogen starvation. Well, you can take that oil droplet and convert it into biodiesel. Right now it's costing about $10 a gallon to make the biodiesel, which is a little more than I want to pay for, a gallon of fuel. But if we could learn how to constitutively turn on the oil production in those algae, then we could easily half that. So we would get down to $5 a gallon on biodiesel.
Do you have to buy it, or could you just feed your algae?
It has to be chemically converted. So you can talk to Dr. Bracco on how to do that. He tries to tell me, and just my brain shuts down. The man's brilliant. But, sometimes we don't speak the same languages. I've been invited to give a talk at a place this summer, and I plan on going in and hitting them with something like, strong immunology, just to show them the difference in the vocabulary that we use. I don't speak their jargon, they don't speak mine. Yeah. Anybody know what dioxic growth is? I'm sorry? Okay, so now define that for me. See, I get that on discussion questions a lot, where you'll take the question and you turn it around, so thinking that that's the answer.
That's the one where it grows on one food, and then that food runs out, and then it kind of levels out, and then it's like, oh, something else I'm going to eat, and then it can grow.
I've asked that question in a lot of master's thesis defenses, and just explain dioxic growth to me. That was a good answer. Very rarely do the students remember that. It catches them off guard for some reason. But once I get them started, they pick up, oh, dioxic growth, two-phase growth. Di-2-oxic phase. So you've got two-phase growth. It's going to use the glucose first. When it runs out of its preferred substrate, it's going to switch over and use the non-preferred substrate.
It's
See?
like when I'm gonna eat a bowl of cereal in the morning, and I see that, I like the Cheerios first, but I'm out of Cheerios, so then I'm gonna eat, the Kix.
I used to like kids.
The conversations we have in here. Y'all are great. You really are. All right. So there's what you're going to get is glucose is going to be exhausted, and that gives you kind of a second lag period. It's the stationary from the glucose, but it's the lag period for the next substrate. Now, if you go in and you add a bunch, an excess of glucose and an excess of lactose, you won't get that two-phase growth because it's only going to use the glucose until it kills itself. Inside a test tube, the bacteria will produce a lot of acids and alcohols and things like that, and they become detrimental.
What do the two lines mean on there? I mean, I know they mean, but.
The blue line is the relative level of beta-galactosidase. So this was a favorite torture method my major professor had. He would make me do these growth curves, and then you would hit the bacteria with some sort of repressor so it slows the growth down to extend the torture that I have. And then I had to do growth curves, but I also had to take samples at every hour point and run enzyme analysis on those samples. So my first semester in graduate school, I spent every other day 24 hours doing an experiment. Yeah. He did that the first semester because he knew after a year I would have said no. That's how we used to do it. That's how you determine the level of enzyme production that was there. We just got a paper published on that. All right. There's cyclic AMP and CRP. CRP is kind of the binding site, catabolite repression site, and a cyclic AMP binds to that region. Cyclic AMP receptor protein, CRP, is the activator protein that the cyclic AMP binds to. It's regulatory nucleotide. It's pretty simple. All right, so you're going to have to look up at the top. At the very top, you see the CRP protein, and cyclic AMP comes in and binds to that protein, which allows it to then bind to the active site there. So the DNA polymerase, the CRP, that's the C on that gene. Let's see. It's up here. There's a C right above where it says active repressor binds. So you get the cyclic AMP binding to the CRP protein. It can bind to that C location right before the promoter. That allows the RNA polymerase to bind to the promoter site, and then you get activation of the LAC-ZYA.
The CAMP binds to the what site?
It's right above here. There's a C site on there. That's they call that the CAP cyclic AMP binding site. So the CAP binding site is on the DNA. The CRP is the protein that the cyclic AMP binds to. That binds to the CAP binding site, and then you have the the RNA polymerase that can then bind to the promoter region.
So what's binding to the CRP protein? What are those called?
Cyclic AMP.
Oh, okay. And then when you put those together, then it's CAMP, right? Or,
No, it's an activated CRP.
oh, okay.
Okay. I know this is a lot of terminology. Let me think. Cyclic AMP is only going to be produced when? When there's no glucose. So when the energy levels are low in the cell, then we want to turn on these non-preferred substrates. So that's the main point you need to get out of this, is in order to regulate the proteins that are being produced, we have multiple ways of doing that. Catabolite repression is one of them. we repress the catabolite from binding to this region by controlling cyclic AMP levels. So if we've got lots of energy from glucose coming into the cell, it blocks that.
So lac operon is an example of catabolite repression. PTS.
That goes back, it all kind of ties in together. Global control of the lac operon. There's a lot of stuff going on in that. I don't think we have time in this class to go into it. If this were a microbial genetics class, I would spend days and days talking about this, but that would be so that you could kind of get the concept.
What do you mean when you say global control?
That's like with a Regulon. Cyclic AMP would be a global controlling molecule.
So it can control everything. Is that what?
It controls things all over the world, but not everything all over the world. Just certain things. Does that make sense?
Like you're controlling everybody's radio in the entire world,
Yeah.
but not
Okay.
the TV.
But not the TVs. So that might be under a separate global regulator of some sort. Remember, the bacterial cells are much smaller, so global control is much easier to handle if your world's that small.
So you could have global control for bacteria.
Flagella, genetics of flagella are pretty highly regulated because the flagella is going to cause the bacterial cell to swim toward or away from things. So we have two directions. We can go in a straight line or we can tumble. Okay, so if that's all you can do, it's like some of those toy cars I had as a kid. If I turned it one way, the thing would roll over. I turned it the other way, it'd go in a straight line. So if you've only got those two controls, getting from point A to point B requires a combination of going back and forth to get there. But bacteria are going to control their flagella genes through catabolite repression. If there's plenty of nutrients, there's no need to swim. So I don't want to turn the flagella on. Or if I'm a biofilm bacteria, then I need to stay stuck in one location. I'm not talking about archaea, so we'll skip that. There's lots of different regulatory systems. The two-component signal regulatory system, I wrote a review article with like 200 references in it when I was in graduate school. So I really don't like that system that much. It's basically a bunch of proteins near the membrane, and it's activated by calcium, or it can activate calcium. So in eukaryotic cells, you can get the two-component system working. There's a lot.
In general, we just call it a two-component regulatory.
Yeah, you don't really even have to know two-component systems for this test. I won't ask a question on it. I am going to talk about quorum sensing. I don't think I'll talk too much about the stringent response except a quick definition of it. So let's see if we can get to, I want to go past chemotaxis. No time. Let's talk about quorum sensing because I think it's cool. General micro class touches on the basics. We can't even go into depth. If you want to become a microbiologist, you'll take 20 classes in order to get all of the stuff that's in this one book. It's just encyclopedic. All right, quorum sensing. Quorum sensing, you reach a certain number, you turn on a gene. It was first discovered, I told you about the stealth squid on the last test. Do you remember that? They grow bacteria that produce light, but the bacteria won't produce light until they reach a certain cell density. So the squid have a light organ, which is tissue that's basically transparent, which is pretty cool in itself. And they grow the bacteria over the day, and then at night, they've reached their quorum and they start producing light. So this is a partnership between the bacterium and the squid.
So the bacteria live in the light organ, correct?
Yes. And the squid feeds sugars and other foods to the light organ to grow the bacteria at specific rates. On a full moon, the amount of light produced exactly equals the amount of light hitting the water. Okay, on a new moon, they have toned it down. It's not so bright. They want to really just obscure their own shadows. And the bobtail squid from Hawaii is the ones that they use because they're kind of small and they're easy to use in research. So quorum sensing kind of ensures the right number of bacteria. I told you about the ship that was illegally dumping sewage because it cost money. So they were dumping sewage and traveling along. Well, the Fisheries Department or USDA or Coast Guard or whoever it was able to follow the ship because the water was starting to glow behind them. And they could follow the trail where they'd been, just like it drawn a line to them. There are bioluminescent bacteria that are found in terrestrial habitats. I told you I've got a cousin in Ohatchee that dug up a log with his new tractor. You get a new toy, you got to dig it up. I'm from Ohatchee, so yeah. And then he calls me and he thought that aliens had landed. He didn't know what the hell was going on. He had a chunk of what he thought was radioactive wood on the back of his four-wheeler driving around showing it to his neighbors.
How often do you get an emergency phone call from a relative about, like, aliens?
Not very often. I think that was probably a first and only time. Although with my relatives, it would not surprise me to have more phone calls about aliens. What got me is he thought it was radioactive, and yet he put it on the back of his four-wheeler and drove around all of his neighbors. If I thought it was radioactive, I'm in the next county calling for help. You know, somebody tell me what's going on. But he showed it off. Look at this. God, I love him. He's one of those good old boys who'll do anything in the world for you. But it was glowing a green glow, kind of like those necklaces you get at Six Flags. You know, that kind of glowing mechanism.
How did the bacteria know not to start glowing, like, during the daytime and they, like, glow at night? You know?
You got to reach that quorum, and it requires oxygen. So in the case of squid, well, in the case of the log was buried and oxygen was being used up so fast, it wasn't glowing. But when he dug it up, the bacteria got enough oxygen that it allowed them to glow. In the case of the squid, they actually secrete oxygen into the organ and control the rate of bioluminescence that way.
Is that how they control how bright it is?
It's a combination of spitting out extra and the amount of oxygen that they feed.
The squid knows, or is that when the squid knows, or the bacteria knows the bright?
Physiologically, somehow, there is some sort of control mechanism. And that's where neurobiologists are going in and trying to study how do they detect the amount of light? How do they control this? It's still kind of a process being studied. It's really cool. Now, how does that affect, let's see, toxin production in pathogenic bacteria. That's how it affects human health. Staphylococcus aureus gets into the system. There may be 100 cells in a clump floating around. They will find a nook or a crevice inside muscle or inside tissue, and they will wall themselves off. White blood cells are going to try to kill them, and they secrete chemicals that cause a clot to form. It's going to kill the white blood cells. So they're surrounded with white blood cells. They're hiding out in the middle, and they just eat the white blood cells slowly over time. Now, as they eat them, they grow in numbers. When they get up where they feel like they're big and bad enough, they break out. They release a kinase later on that breaks the clot. So you have a coagulase that's turned on early in the growth cycle of the bacteria. But you have a kinase that's under some sort of quorum sensing control that's turned on later in the process. And there's all sorts of toxin genes that are done that way. They're only produced when the bacteria reaches a certain quorum level.
So is it kind of go undetected other than since can they figure out something?
It can. It can. There's a guy that works on our autoclaves. Went in for eye surgery. Had eye surgery. Staph aureus got into his bloodstream. and he had torn tendons in his shoulder when he was younger, so it tends to stick to scar tissue. And it started a staph infection in his shoulder. And he was complaining of pain in his shoulder. Then he kept going to the doctors, and they're like, giving him, take aspirin, they just weren't catching it. And he's like, no, you don't understand. I'm hurting here. Something's wrong. So he finally got the right scan, and they called him back to the doctor's office and did surgery immediately on him. Staph infections are scary. You've heard of MRSA, the methicillin-resistant Staph aureus. That's becoming a major ordeal. There's high school athlete, football player, about 3 years ago, got a staph infection, and they wound up amputating both his legs from staph infections. So they used to have problems. They called it community-acquired staph aureus. It's now all dumped into that methicillin-resistant staph aureus group. But they called it spider bite syndrome in local prisons and jails. The prisoners were complaining that spiders were biting them at night because there'd be two little holes open up where the staph aureus kind of vent themselves. Nobody knows how or why those two holes show up, but it looked like spider bites on the body, and it
What
turns
if
out.
they know that they're trying to look like spider bites?
Huh?
What if the bacteria, what if they know?
Okay, now we need to write a book about that. But yeah, I don't think there's any anthropomorphic knowledge in the bacteria. I think it's got a physiological reason behind it. But what happened is in the jail systems, they were able to reduce that by about 90% by giving them a squirt bottle with 10% chlorine bleach in it and telling them to clean up their cells. That reduced it a lot, except for the ones that were squirting it in each other's eyes and that sort of thing. And then, just let them have staph.
How do I avoid getting this infection? Like, that's
Wash your hands. If you get a cut, you can use triple antibiotic cream on the cut, as long as it's not like internal. Don't shove it up your nose. Just wash your hands. Use triple antibiotic creams on cuts. Don't go around barefoot outside, especially if you're immunocompromised. I'm a diabetic, so I always wear shoes, house shoes or something. I have sandals I wear at home all the time because you want to cut down on any kind of cuts. If you're working with patients, you need to be fastidious and obsessive about washing your hands and being clean. I found out working with my mom this past weekend that I am not cut out for that type of health care. It's just not. Some people are saints. Bless your hearts if you're going into that kind of work. I don't want to do it. There's just, like I say, there's some things a son's not supposed to see on his mother. Get burned into the brain. Oh, doggone it. So we got quorum sensing. Read about quorum sensing. It's really cool. And there's a good chapter there. Let's make sure.
is quorum sensitive. Is it in the book in that?
Yeah, it goes into a lot more detail. Read about quorum sensing. And, gosh, I wish I
One
had.
more thing to see on that time.
That's all we're gonna have time for. It looks like we're running out of time. Quorum sensing. The only other thing I would be interested in, just for your information, I won't put it on the test, is heat shock proteins are pretty cool. Heat shock proteins are really cool.
So the rest of that's right. Next step.
That's all we, yeah. No, I probably won't have time to even cover it. We'll move on.
Is the attenuation going to be on there, or do we already?